A warm welcome and welcome back to the Code of Life OpenHBI course. Today we want to talk about bugs in the source code, so to say. We are talking about genetic changes. First of all, we need to distinguish two terms here. The first one is a genetic variant. A genetic variant is a polymorphism within a genetic code. That means nothing more than there's an individual representation in a genetic code. Not all of the variants are necessarily mutations. We refer to mutations once a variant has a measurable impact in the products. Mutations typically occur spontaneously and undirected. And therefore, these mutations are important to understand because they change the function of the cells, the products of the cell, or even produce something completely different. However, these mutations are considered as the foundation for evolution because by changing and altering some functions in the cells, it's the only way to play around and see how does the function of the body of the organism changes. And typically the best adapted species survive, so therefore some mutations are very important over time. Mutations are triggered by so-called mutagenes, and these mutagenes are any kind of components affecting the genetic material. For instance, any kind of radiation. It's X-ray, it's electromagnetic, it's sunbeam, and so on. Chemicals, temperature, high pressure, they are all referred to mutagenes because they can have impact on the DNA, as this is a really sensitive structure. And you see here on the right-hand side the international symbol for any kind of mutagenes. Some understanding about mutations. We can distinguish mutations into different levels of mutations. First of all, we can have mutations in the germline cells. You can remember germline cells are important for reproducing organisms. And if you have any kind of mutations in the germline, then you can be sure that these changes are inheritable. That means any body, any person which is coming after this body is really affected by this change. Somatic mutations are only affecting the current individual. This is important because a lot of these mutations are acquired during lifetimes, and these mutations have only impact on one individual and not on a whole line of family members. Where are locations for mutations? And this is really only an excerpt, but you have different levels. Of course, on the lowest level, it could be on a gene, so certain regions. On a chromosome level, you have certain chromosome parts which are affected, or on a genome level. We want to deep dive a little bit here and understand which kind of mutations are available. First of all, we have these gene mutations. Typically, they are single nucleotide polymorphisms, so-called SNPs. They affect really a single locus, and this is changing. For instance, A is replaced by a T or a C or whatever on the genetic level. Then you have so-called indels, insertions, or deletions. That means an arbitrary number of nucleobases is changed. Either they are removed or inserted in addition. That means the DNA gets longer or shorter at that period of time. We can distinguish these mutations into non-functional, which is important because due to the amino acid coding site, non-functional mutations have no impact on the products. Typically, these are SNPs. That means if you change or alter the third nucleobase in the amino acid, and this is redundant due to the amino acid coding site, then you have really a non-functional mutation. Functional mutations, and this is important, they typically impact the products of the DNA and RNA which is produced by the ribosomes. And there are different alternatives how they could impact this. A so-called missense mutation changes a triplet that there is another amino acid produced instead of the original one. That means you replace one amino acid by another. That means the protein looks slightly different. However, it's still the same protein. The next kind of mutation is the nonsense mutation. This converts typically any kind of triplet to a so-called stop codon. That means although there's a lot of coding information, there's a stop codon at an earlier stage preventing to produce the whole protein. It's really a subsequent issue because you have only a small part of the protein built. Maybe the most important information is never built into the protein. And then there is, last but not least, the so-called non-stop mutation. Non-stop mutations change any kind of stop codon into any other amino acid. That means instead of stopping the coding at a certain region, it still produces a longer and longer and longer amino acid sequence. And this is really a challenge because you get a more complex protein which is also typically not capable to produce the estimated effect. So what is the next level? The next level are chromosome mutations. On chromosome level, you have also these kind of indel mutations. Indel here means that certain parts from other chromosomes are copied or replicated into the chromosome. That means the chromosome gets larger or smaller in terms of the deletion. You have duplication. That means certain areas on the chromosome are duplicated multiple times. This is also important in the terms of plants, because by multiplying certain regions in chromosomes, you can have a higher growth rate or less water consumption and all these things. However, in humans, it definitely changes some kinds of functions. This is important. Then you have so-called inversion mutations. That means a certain segment is inverted due to the DNA synthesis at that point. You have translocations. That means certain regions from one chromosome are exchanged with another chromosome. That means you can see that certain parts of a chromosome A are moved to chromosome B and vice versa. You have transpositions where segments are added to other chromosomes and so on. And you see it's just the next level. You have the gene level, now you have the chromosome level, and last but not least you have also the genome level on the whole genome. This is typically resulting in another category of mutations because they affect the total number of chromosomes in the body. Typically, you know, humans have a diploid chromosome set. And these chromosome mutations, these genome mutations result in multiple versions of the same chromosome. So you see examples for that in trisomy 21. And what happens here is that one chromosome is replicated not only once, but it's replicated twice. That means you have 3 times one chromosome, and this has always an impact on the structure of the information which is responsible and stored in this chromosome. It's typically exaggerated here, that you have more information produced out of these chromosomes because you have more source code responsible for the same material. What to take home from this lecture? It's important to understand not every variation is necessarily a mutation. Mutations are occurring spontaneously and undirected on different levels, on gene level, on chromosome level, and on the whole genome level. And it's very important also to understand that these mutations are not necessarily always pathogenic, so they are not always bad. They are important to increase the variability and the ability to adapt to certain environmental settings. So they are also the foundation for evaluation. And you see, this is also the understanding that you have to know that certain kinds of mutations are very important in disease development. For instance, our cancer use case we introduced at the beginning. So how to detect these individual mutations is a really challenging task to process these data within a short period of time. So next time we will talk about how to use the data and how to create the data. So stay tuned and see you next time.